Test crosses and linkage. Suppose you're quite hungry and you start thinking about your favorite food, watermelon. Wow, but how do we get the perfect watermelon? Some days the perfect one might be the biggest one possible, and we can eat as much as we want. Other days, the perfect one might be the cheapest one possible, so we can spend the money saved on other stuff. So how can we consistently create watermelons accordingly with our desires? That's where genes come in. Suppose there's a gene, the small gene, denoted by S, which controls for size. Genes are usually named by their mutant phenotype. Thus, the phenotype of a watermelon with mutated small genes is small. Let's also say that the small mutation is recessive, meaning both copies of the small gene must be mutated in order for the mutant phenotype to show. Now, if we're given a random big watermelon, we can find out what the watermelon's genotype is, or what kind of small gene it carries, through what's called a test cross. We breed the big watermelon with a small watermelon, which we know has to carry 2 mutated copies of the small gene, and look at phenotypes of the offspring, also known as the first filial generation, or F1 generation, to determine the genotype of the big watermelon. If all the offspring in F1 generation are big, then the big watermelon must carry 2 wild-type small genes. If half of the offspring are big and half of the offspring are small, then the big watermelon must carry a wild-type copy of the small gene and a mutated copy of the small gene. Now, what if we have 2 genes, one being the small gene still, and a new one called the blue gene? Let's start with a big red watermelon we know that has genotype big S, and big B, and a small blue watermelon with genotype small s, and small b. When we look at the expected offspring for a cross between the 2 watermelons, we need to consider all possible combinations between the small and blue genes. However, notice that for this, and this, we get big S, big B for all of these. Similarly, for the small blue watermelon, we would get small s and small b for all possible combinations. Thus, the expected offspring would all get 1 copy of a wild-type small gene, a mutated small gene, a wild-type blue gene, and a mutated blue gene. So, they'd all look big and red. Then, when we cross any 2 watermelons from the F1 generation, we can see this time there are 4 combinations of small and blue genes: big S, big B, big S, small s, big B, and small s, small b. Thus, we get 4 times 4 equals 16 possible genotypes in the second filial generation, or F2 generation. We can color code by the phenotypes of the genotypes, and we get a 9:3 to 3:1 ratio of possible phenotypes. Linkage occurs when 2 genes are physically close to each other on the same chromosome, so that when meiosis occurs, the probability of a crossover event occurring between the 2 genes is a lot less likely. Here, you can see is the place where a crossover event must occur for these 2 color genes to recombine, while here is where a crossover event can occur. We can actually estimate how close these genes are to each other, which we'll do later. Now, to test for linkage, we can test cross an F1 offspring to a double mutant. We would expect these genotypes, which we can color code by their phenotypes, and we get a 1:1:1:1 ratio of phenotypes. Suppose instead we get 450 big red watermelons, 50 big blue watermelons, 50 small red watermelons, and 450 small blue watermelons in this test cross. This is in a 9:1:1:9 ratio, not the expected 1:1:1:1 ratio. We can calculate the recombination frequency by dividing the number of recombinants, which are the watermelons with phenotypes different than the parental ones. Remember, we started with big red watermelon and small blue watermelon. So we divide the number of recombinants, big blue watermelon and small red watermelon, by the total number of offspring in the F2 generation to get our recombination frequency. So 50 plus 50 over 450 plus 50 plus 50 plus 450 is equal to 100 over 1,000, which is also equal to 10% recombination frequency. Each 1% recombination frequency is equal to 1 centimorgan, so the blue gene and the small gene are 10 centimorgans apart. To double check our results, 10% recombination frequency means 90% of the time we get parental combinations, big S big B and small s small b, and 10% of the times we get recombinations, big S small b and small s big B. Big S big B is passed down 90 divided by 2 equals 45% of the time, and so on and so on. Since we're test crossing with a small blue watermelon that must pass down small s small b, the combination of a small s small b for the small blue watermelon occurs 100% of the time. We can again color code by phenotype, and we can see the proportion of phenotypes that we would expect for linked genes that are 10 centimorgans apart. For 1,000 watermelons, we would expect 450 big red watermelons, 50 big blue watermelons, 50 small red watermelons, and 450 small blue watermelons. This is consistent with what we started with. Thanks for watching, and here's a watermelon.